in this part, after this introduction of phytoplankton biology and biomonitoring in lakes by Frédéric, I will talk about how we developed the metabarcoding part for this. Soon. It's not. I think I will use the other. I don't know. Okay, I will do this like that. So just to show you, now you start to know this workflow by heart, that we are back at this point where we don't know precisely how to sample, how to extract DNA, which barcode we are going to use, and how to analyze the data. The only things we know is that we wanted to use Illumina sequencing because it's the most interesting for metabarcoding. And so we first started to ask ourselves, which barcode are we going to use for this phytoplankton metabarcoding? So again, what we wanted ideally is a barcode that would be present in every phytoplankton. And Frédéric showed us that it's a very, very diverse group, including prokaryotes, bacteria, cyanobacteria, and eukaryotes. We wanted this barcode to be specific to phytoplankton and not, for instance, having heterotrophic bacteria. And we wanted it to be variable enough to distinguish different species of algae. And we wanted to be of a maximum size of 450 bare page, which is the sequencing size of Illumina, and also to be represented in reference databases. So this was the objective. And how did we choose this barcode? We first, as for diatoms, made a selection of the interesting marker genes that could be used for phytoplankton. And then in a second time, we selected primers that were present in these different marker genes. And we tested the performance of these primers to see how they really works. So the interesting marker genes are always the same. We can have nuclear DNA, the ribosomal gene that is present in the nuclear DNA of 18S. Or we can also target plastid DNA present in the chloroplast, such as ribosomal gene 16S, ribosomal gene 23S. This one codes for the large subunit of the ribosomes, or other genes such as TUFA or RBCL that were used for the diatoms. And we could have also investigated mitochondrial genes, but for algae, they are not so well interesting. So what are the advantages of nuclear DNA? It's that they are present in reference databases, as it was also a good point when we investigated diatom metabarcoding. We have those two databases, PR2 and Silva, that are often used in ecological studies. Ribosomal genes are very interesting because they have those highly variable regions flanked by conserved regions. But unfortunately, they are not universal because they won't be present in cyanobacteria. 18S ribosomal gene is only present in nuclear DNA of eukaryotes. So we won't target cyanobacteria with this gene. And also, as Frédéric showed yesterday, they are not variable enough for some species as diatoms. And their length, if you remember my talk yesterday, their length could change between some taxa. And so we couldn't have access to those sequences. Then, what about plastid genes? So the first one, RBCL, was really nice for diatoms. So, but what about phytoplankton? So they are universal. They will be present in cyanobacteria and eukaryotic phytoplankton. They are variable enough for species detection, and they are recommended for plant detection. But there are few reference available for some algal group. Now they have good reference for diatoms, but it's not the case for all the algal groups. And we cannot design also primers for all the algal groups for this gene. They must be specific to each algal class, which is not interesting for metabarcoding, optimal for metabarcoding. Another gene, TUF-A, so same, it's universal, present in cyanobacteria and eukaryotic phytoplankton, and it can be variable enough for species and even subspecies detection. But again, we have very few reference databases available, and same as RBCL, it is complex to design primers for the global diversity of the phytoplankton. And now we have the ribosomal genes of the chloroplast and that are also present in cyanobacteria. And they are much more interested because they are universal. They have very good reference for cyanobacteria, particularly for the 16S, because it's really the barcode that is used to target bacterial diversity in general. These genes has highly variable regions and conserved regions as every ribosomal genes. And there are a lot of primers already developed in literature for microalgal diversity. And the other ribosomal genes that is present in the chloroplast and also cyanobacteria is the 23S genes, which present the same quality as the 16S, but the reference are still a little bit poor for phytoplankton. But on contrary, it presents a higher phylogenetic resolution than the 16S. So that after this quick overview of all the universal marker genes, we decided to test to do phytoplankton metabarcoding uses those two genes because of all the advantages I discussed a little bit earlier. So the next step is to find primers. So either you can find primers of those genes directly from literature designed by a previous team, or you can try to design your primers. And it's what we did in the lab. And we took from literature and from primers that we designed, we gathered 2 candidate primers for the phytoplankton metabarcoding. And then the next step is to try the performance of those primers first in silico. That means using reference libraries to see the performance of these primers and to test different things that I will detail later. And then after this first selection, we tested the performance of those primers in practice using mock communities of non-phytoplankton composition to make at the end a final selection. So with in silico analysis, we can really test a broad diversity of things in our primers. The first one is using oligoanalyzer. It's a tool that can test if your primers are adapted to PCR, because maybe they can target all the diversity, but if they don't perform well during PCR, it's not good primers. So For instance, one thing we have to be aware is the temperature of hybridation of those primers. During PCR, after the denaturation of DNA, you anneal your primers, and this will be done at different temperatures depending on the nucleotide composition. As Agnès said in the beginning of the workshop, there are 3 boundaries, hydrogen boundaries for between G and C, and 2 only between A and T. And this will influence the optimal hybridation temperature of your primer. And what you want is that the forward primer and the reverse primer has a similar optimal temperature of hybridation. So you can test this. And then also you can check if you can have cross-dimmer. That means that your 2 primers will hybrid together. And if they do that, they won't hybrid on the template, and it will be complex to amplify your DNA. Another thing you don't want is that your primer hybrid on itself, like doing this hairpin, and you can test that with this kind of tool, oligoanalyzer. Then the second thing we tested in silico is the amplification efficiency, that is precisely to look how many sequence match with my primers, just to see how many sequence have the complementary regions of my primers. Another thing that Frédéric told about for diatoms is to see the resolutions of these primers, how many species they will help to define. And then it will be also to look if your primers are specific to the phytoplankton, or how many unwanted species you will get with your primers. And then also we tested the in silico assignation efficiency, that is if we can retrieve a taxonomic assignation at the end with the barcode, and the percentage of amplicons that are correctly assigned to their corresponding taxonomy. So after this world work, we could find some interesting candidates. Starting from those 12 couples of primers, we found 5 interesting couples of primers, 2 from the 16S RNA genes and 3 for the 23S RNA gene. They were all good for PCR. They all have good resolution, even if taxonomic resolution, even if this taxonomic resolution was better for 23S. And they had all good specificity, except for one 16S couple of primers that amplified heterotrophic bacteria. And we found it interesting to keep these primers during our next test as a comparative. So now we have our first selection of 5 primers, and now we are going to look how they perform in real conditions using mock communities. So we did the first mock community with 10 strains from the culture collection of our lab. We cultivate the phytoplankton, the microalgae. We extracted DNA. We put this DNA in equimolar concentration. We amplify this DNA with PCR in triplicates. We sequence with Illumina, and we recovered ASV at the end with Data2 pipelines. So it's to see who were the phytoplankton we looked at for this first mock community. So we tried to took phytoplankton from a very diverse group of microalgae that you can have here. And the results are here. So first, one question is, using those 5 primers that you have here in the x-axis, are we able to detect the 10 control species we put in the mock community? And the results are here. For the 23S primers, we were able to detect all the species we put in the mock community. And for the 16S, it was a little less optimal. And then we wanted to see the percentage of ASV that were assigned to the species we actually put in the mock community. And we see that that we have mitigate results for the 23S, very good results for one 16S, a couple of primers, and very bad results for another couple of 16S primers. But the number of ASV is not representative of the number of reads. This is quite technical. I don't know if I made myself clear. You sequence, and then those sequence you get ASV, which are sequences corrected from their sequencing errors. But you can have ASV with very dominant, that represent very dominant microalgae with 80, I don't know, 1,000 of sequence, and you can have ASV with only one sequence, for instance. So it's not you don't have the same information. And if we look at the number of reads that were affiliated to control species of the mock community one, we see that we almost 100% of the reads were indeed affiliated to the algae we put in the mock community. I don't know if I was clear for this part. It's quite, but we can discuss of this a little bit later. So based on those results, we can see that with our 323 primers, we really get good results. And we decided to select this one that presents the highest results. And then for the 16S, one primer, no, I didn't say that, and it's very interesting, is that here for the second 16S primer, we really got less than 20% of the reads that were affiliated to algae. That means that the other percentage of the reads were affiliated to other things and mainly heterotrophic bacteria. So it was this primer that is not very specific for microalgae. So we decided of those two couple of primers, and then we we tested them with the second mock communities, and for the second mock community, we increased the number of species and the diversity we wanted to test, and we did the same things with retrieving ASV at the end with a data tree pipeline. So here are the new species we put in this mock community too. And here are the results. So you have all the species that were put in this mock community, and you have the one that were recovered in the 16S with the 16S primers, and the ones that were recovered with the 23S primers. And we could retrieve almost all the species that we put in mock community 2, except for the 16S primers that didn't have sequence of Hematococcus lacustris that was in the mock community. And in color, you have if those sequences were well assigned or not directly using reference libraries. And we see that we really retrieve very well the taxonomic affiliation with 23S, and that it was less optimal for 16S. And then we as we put DNA in equimolar concentration, we can see for all the species the number of reads that were retrieved. And we see that for the 23S, this equimolar concentration were better retrieved than for the 16S. So overall, this mock community analysis showed that the 23S primers perform better to recover the whole 18 species that were put, to have a better assignation at the species level at the end, and to give more homogeneous abundance at the end. And so it's how we choose our primers for phytoplankton metabarcoding analysis. And normally there should be, but I think we are a little bit in advance. There should be a coffee break at this moment. No. I have 20 minutes because you ended before. No. But I can continue. There is no problem. Maybe if there are some questions, yeah. Maybe there are some parts that I was not very clear.
It seems to me that the audience is quite familiar with this topic. Everything is clear.
They know everything. So if you are okay, we can continue and see all the other steps for this phytoplankton metabarcoding. Yeah. Okay.
Question from the Zoom. On slide mock community 2, the graphs on the right side, 16S and 23S, what represents light gray and what dark gray color?
Yeah, it's because I used a graph that I already produced, and you will see in the second part of my talk that during this mock community analysis, we also tried different DNA extraction protocols. So, but I was not going to speak about that at this time, but I will speak of them later. Often, as many each steps can be influenced by DNA extraction and everything. When we do this kind of test with control species, we try different. It's a good way to try different things, and that's why we have those two colors. Yeah.
Thank you a lot.
So not time for a break. And we will see for the sampling. So as we are interested in seeing the phytoplankton metabarcoding for biomonitoring, we wanted our sampling to be easily done by a private office everywhere in France to sample this phytoplankton. So the protocol had to be adapted for biomonitoring. So simple, not time-consuming in the field, and that use a minimum of material easily provided to people that are actually doing the sampling. And this material has to be clean, and but I will describe the protocol later, but we use those Sterivex filters that are indeed quite expensive, but very simple to use in the field. And we decided to conserve the DNA using a lysis buffer because having a freezer in the field is not the case for everyone. So basically, how we sampled this phytoplankton for DNA metabarcoding, it's you take water with a bucket, and then you you take water with a syringe of 50 milliliters. I don't know if you can see well in the back of the room. And then you put the Sterivex filter at the end of the syringe, and you press the syringe to filter water. And we decided to choose 250 milliliters of water to be filtrated. So you redo this operation in order to have filtrated 250 milliliters. And then when it's done, you put a little cap at the end of the filter, at one end of the filter, and you add the lysis buffer, 2 milliliters of lysis buffer to the Sterivex in order that the filter will be totally covered by the solution. And then we close the Sterivex with another cap, the other side of the Sterivex, and you put your Sterivex in a bag, and it's okay. So it doesn't take so much time on the field, which is nice. And then I will talk about DNA extraction, how we tested different protocols. So those protocols had to be adapted to a Sterivex filled with lysis buffer. And we decided to do the lysis step when we break down the cells and everything directly into the Sterivex. So there is a vortex step to detach the cells from the filter, and then we use detergent, SDS, and enzymes to do the cell lysis. And then we tested different protocols to see how they perform. So precisely in this little test, we compared 2 DNA protocol, the one that is a very easy one, Genilute, that was previously used in the lab, and nucleospin soil that Frederic told about, that was also already used in the lab, and that gave good results for diatom DNA extraction. And we also tested 2 filtration methods, because in our lab we routinely do filtration for the Alpine Lake biomonitoring on filters. And we wanted to see the impact of the filtration method in the results at the end. And this test was done at the time we were tested mock community 1, so we had those 5 primers. So here you have the study design. We took a sample from the lake right in front of our lab, and we either took one sample for microscopy analysis, either filtrate onto a Sterivex and extract DNA with nucleospin soil kits, or either filtrate on a filter and use the same extraction protocol, and filtrate on the filter and use the other DNA extraction protocol. And here are the results we got from this study. So you have here all the samples that were tested for. So you have many samples because you have different 16S and 23S primers that were tested. And you have the composition in the main families of phytoplankton for all the samples. And they are clustered based on their similarity. And here you have the microscopy, the floristic list from the microscopy. And what we can see, on the top, you have all the 16S primers, and on the bottom, you have all the results obtained with the 23S primer, regardless of the DNA extraction and the filtration technique. And we see that we were closer to the microscopic list with the 23S. And this was mainly due to poor taxonomic assignation with the 16S, and particularly this blue color are unclassified bacillariophyta that could be classified with the 23S. Then if we look a little bit closer for the 16S primers, we see that here you have one primers and the three different filtration and DNA extraction we used. And here you have the other primers. Really, the primers had an influence on the diversity obtained. And it's mainly due because those primers, this couple of primers amplified a lot of what is called unclassified and are in fact heterotrophic bacteria. So these primers was the one that was not specific to phytoplankton. And at the end, we see that indeed majority of the sequence are heterotrophic bacteria. And if we look for the 23S primers, we see that what most influenced was in fact the DNA extraction protocol and the filtration methods. The sample doesn't group based on the primers used, but more on the DNA extraction protocol and the filtration method. It reminds me of Agnes showed you right before. Those differences are not that major, but it can be good if you have a survey to keep the same protocol to really compare your sample together. Then we did another test at the time of the mock community 2, and we tested 4 different protocols from Genelute, where we centrifugate so at this time we were with a culture of microalgae, the 18 culture of microalgae I presented you for the mock community 2. And we centrifugate this culture and did the extraction, or we filtrated this culture and did the extraction. We also tried another protocol that is based on directly selection of cells under the microscope. And we also tested, because we had this in our lab, a robot that is a automated DNA extraction procedure that can be interesting when you have large quantities of DNA extraction to do. But it's quite expensive, so so what we saw here, you have the results for the automat, the shellac, this one, the Genelute with filtration and Genelute without filtration. And we see that we best recover the 18 species with the automat and the Genelute protocol without filtration. And also, the automat presented a higher DNA quantity and quality at the end of the extraction. Plus, it is way more faster and gives more reproducible results in a routine use. So we decided, but it's specific in our lab because not everyone has an automat for DNA extraction, but we decided that it will be used for phytoplankton biomonitoring. And if you are ready before the coffee break, yeah, maybe. And then bioinformatics part. Okay. Okay, so that's okay. And also you can easily compare different data sets, because when you reconstruct OTUs, you put a similarities threshold. That means that you have to redefine OTUs each time you input a new sequence. With Data2, it's not mandatory, so it's really a nice pipeline for bioinformatic analysis. And also it has this nice website that describes all the pipeline steps, etc., etc. So basically, what are all the different steps in details? You received your FASTQ files from Illumina platform with files from read 1 and files from read 2. Here I only put 1, but if you have 20 samples, you will have 20 files for read 1 and 20 files for read 2. And then the first step we did is to remove the primer sequences. And then we did a quality filtering of your reads. And then we go through the data2 pipeline that calculates that try to model the errors and then correct the sequence from those errors. So it's those two steps, and data2 does it by itself. And then it merges the reads, R1 and R2, and then you have your ASV table that can be almost can be used for your analysis. Because before you have to remove chimera. And also data2 add a step to keep ASV of the expecting length to be sure that you have the sequence you wanted and not a specific ASV that can be due to other errors that occurs. And then you can assign taxonomy. And this was mainly, as I told you, mainly done through data2, but we also use the other tools. And for instance, to remove the primer sequence, we use a very famous tool that is cutadapt. And you can find documentation on the internet. Then all those steps are done with data2. And then for the taxonomic assignation, you can do it through data2, and it really works well. But we decide to use Mother, but they really give the same results at the end. So now you have the pipelines. What do you check when you just received your sequence and you want to go through this pipeline? So first, data2 told you to check for the quality. You can check the quality of your sequence. So those graphs are not easy to read, but basically you have all the read one sequence, so around 250 bare paste, and all the read two sequence. And here in green or red, it's the quality score. And in y-axis, you have the quality score of each base of your sequence. And we can see that this quality score is high, almost around all the length of read one, and that it decreases a lot at the end of read two. It's something that is really common and known for Illumina sequencing. At the end, the sequencer makes more mistakes when you arrive at the end of read two. But it's not a problem. You just have to know it. Data2 recommend you to trim those lower quality ends, so you can check where to cut based on your results. If you have very bad results all along the reads, you can send a mail to the sequencing platform and ask what happened, because it's not normal. Then, so the quality filtering is done automatically based on the quality score. Then error models and inferring samples, data2 does it by itself. And then you can merge the reads. So at these steps, you can lose some sequences, but you mustn't lose too many sequences. So to see if your bioinformatic pipelines goes well, you have a sanity check here to see, okay, I lose some sequences, but you mustn't lose too many sequences. For instance, if you lose too many sequences, is maybe because you, for the previous step, you cut too many bases, and at the end, you don't have any overlap. It can be something. Then there is a chimera step. So again, at this step, you can remove a certain amount of sequence, but it must be 50% of your reads. If it is 50% of your reads, that means that you have some troubles somewhere, but often it doesn't happen. And for data 2, they say that you can cut the reads based on the expected length. So for instance, for phytoplankton with the primers we choose for 23S, we're expecting a length of 358 bare page. And this, you can have a look carefully of this, and I will show you how a parameter you put in a bioinformatic pipelines can have consequence at the end for your results. For instance, if we were very strict and we just keep reads that were 358 bare pages long, we could have missed some organisms. And it's represented in that figure. You have different families of phytoplankton. And here you have the length of the sequence we got after the sequencing. So you can see that for most of the families, the length was of 358 bare page nucleotides as expected. But if we were very stringent, we see that there is one chlorophyce here that has a higher length of the amplicon. And it was in fact an interesting chlorophyce because it was Hematococcus lacustris that we are expected to find. So it was an example to show that you may want to check and to be sure of you won't choose this expected length from nowhere. You have to check and be sure that it won't make you lose sequences you're interested in. But it's very rare. You can see that for most of the families, they have this expected length, but it can happen. And then why did we choose mother? It's because when we compared data to assignation algorithm and mother, mother was a little bit better in for the assignation, but it was a very few percentage, so it was just like this. And that's it for this pipeline. And now for the next part with Benjamin, you will see a little bit more the reference library we design in the lab, which is called Phytool. And you will see, which is very interesting for this workshop, how this workflow performs for the phytoplankton by indication. Did it perform well or not? If you have any questions on this very technical part for bioinformatics. Yes.
I have actually questions about the sampling part, about these Stervex filters. What do you think, how much amount of water is good to filter? Because maybe for eutrophic systems, it's okay, 50 milliliters, one syringe, but what about oligotrophic system? And
yeah.
One more question. About lysis buffer. Some authors provided that it's also good to use Longmire solution. Do you have experience with the Longmire solution for the preservation of DNA?
For the second question, for my part, I don't have experience. I don't know if it is the case for Agnès and Frédéric and Benjamin.
You remember from yesterday, the preservation study I showed, the homemade lysis buffer was in fact Longmire solution. And so we could see it performed well, but in Sterivex, I'm not experienced on that.
And in Sterivex, it's something we will try with the students this year. So we are still developing and testing these protocols. And there will be a student that will compare ethanol, this lysis buffer, or freezing samples and everything to see how the DNA concentration diminish and everything. So yeah, for now we use this lysis buffer, but maybe it can be improved.
And also about the volume,
And about
the sampling volume.
The sampling volume, it was based on because for microscopy you use 250 milliliters. No, it was not.
For sure, when you're in a eutrophic lake, you can't filter 250 milliliters. So in the case of the protocols that we send to the private laboratories, we ask them to note the volume they sample if they don't sample 250 milliliters. And the objective is to have some material. And for sure, when you're in a eutrophic lake, the 50 milliliters is really enough. We have enough phytoplankton to do the PCR and so on.
And for oligotrophic lake, because we did in fact the DNA extraction from various lakes everywhere in France, and we had very oligotrophic lakes. We have very less concentration of DNA, but still enough to do the PCR and everything. And as the PCR is very sensible, then you can have your phytoplankton community. So it's yeah.
Other questions? No questions about phytoplankton? Milos? Yes, you have.
To Daniela.
Thank you, Milos.
Okay.
We just want to say to all of you that during this project, we will make different protocols for diatoms, phytoplankton, microphytes. It will be public document, and we will share to everyone. Thank you.
Maybe we can say also that these pictures are in fact from a video that you can access easily, and we will send you the link if you want.
In fact, yes, we produce some very simple protocols for the field workers, and they can even flash. There is a flash code, so you remember the COVID situation, we had to flash. So here they flash, and it's directly going to the YouTube video if they had some problem, but it's not very complicated, in fact.